so today I'll talk about the evolution of robustness. So actually, the original title was Evolution of Robustness and Evolvability. And when I finished to prepare my talk, I realized that it was not too much about evolvability. So I changed the title, and I apologize for people who joined for the evolvability part. But well, we can probably discuss about that afterwards. So, so this is a kind of review summary of my recent and not so recent work about the evolution of genetic architectures. And so this idea of working on the quantitative genetics of robustness, that's something that I've done since now some time. And actually, it's at the core of some modern quantitative genetics question. So the baseline of this is that most biologists consider that living organisms are robust. So this is somehow, it's a strange claim. We won't have time to discuss about this, but if you compare a stone and a beetle, for instance, for some reason, a lot of people consider that the beetle is more robust than the stone. But of course, if you step on them, it's less obvious. So I don't know why it's so common that it's so obvious that organisms are robust, but they are robust in their way, let's say. And so mostly, I mean, the major effect of biological robustness is that the phenotype is under control. So if you take a species of monkey, you give them twice as many bananas as the other ones. I mean, they don't get twice bigger and they don't change completely appearance. And I mean, it's still the same species, right? It's not because you feed twice that you are twice bigger. So that's one aspect of robustness, being able to maintain a specific phenotype independently from environmental conditions. And of course, the other way organisms are robust is their reaction to mutations. So actually, because the talk was broadcasted, I had to remove copyrighted pictures, and I replaced it with AI-generated pictures. So this is what you get when you ask a poster movie about mutants to artificial intelligence algorithms. And actually, it's pretty convincing. I don't know what you think about it, but actually, these guys have real mutant faces. But this happens in Hollywood movies, and you know that in reality, when you mutate genes, most of the time, actually, the mutations are quite silent. You don't see them. And with some important mutations, you might either generate the death of the organism or slight phenotypic differences, but you never get green skin and wings at the same time. I mean, most organisms are quite robust to large-scale mutations. But of course, I mean, organisms are not infinitely robust. And one way this lack of robustness is obvious is phenotypic plasticity. And for instance, I mean, this is a textbook example, right? When you plant the seeds of a plant, they often don't germinate at the same time. It's just because, okay, if all seeds germinate the same day and then there is cold temperature or bad weather conditions or whatever, everyone dies and it's bad for the organism, right? So bed edging is clearly some kind of selective advantage for spreading the phenotype and not being robust. And of course, on the genetic side, if you want to adapt to a changing environment, you need some mutations to have visible effects. So organisms cannot be infinitely robust, right? And okay, when you have large-scale climate change, and this happened to the dinosaurs, and this will probably happen to us soon, you need to get phenotypic variation from genetic changes to be able to adapt to the environment. So Darwinian evolution requires non-robust genetic architectures. So this is the main framework. And then the evolution of robustness has to be understood in this framework. So that's why the question is so complex. So as far as I'm concerned, as a quantitative geneticist, the history of the evolution of robustness roots in Waddington's work. So Waddington was a developmental biologist, and he wrote a lot of interesting text, influential text about evolution of robustness. So about at the same time, a Russian geneticist, Schmolhausen, published very similar ideas, but he didn't get the same success in terms of the same scientific success, and probably because he published in Russian, it doesn't really help. But actually, these ideas were around in the 1940s. So basically, the theory of canalization proposed by Conrad Waddington is a verbal model about the adaptive advantage of being robust in general. So okay, the theory is much more complex than that, and okay, I'm not able to give a seminar about history of science on these specific things. It's very interesting, it's very rich, very confusing as well sometimes. But so the way I could understand naively the theory of canalization is that it's a theory about adaptive robustness. And you all know this famous picture from Waddington that is illustrating somehow the phenotypic robustness. So the ball here, it has to be understood as a trajectory of a cell during development. And each time you have different trajectories, it corresponds to cell differentiation. And the state of the cell is somehow funneled or canalized by this phenotypic, by this epigenetic landscape, forcing the ball to stay here. So you have hills here, and canalization is just the physiological process of keeping the ball in this valley here. So that's what maintains the robustness of the phenotype. And of course, this is just a verbal model, and it's necessary to confirm this with empirical observations and by formal models. And so for some reason, you can see, actually, I'm not sure you can see, I mean, it becomes a side test, sorry about that, but actually, the first theoretical models about, I mean, implementing this idea, as far as I can tell, it dates, I mean, they first arose at the very end of the 20th century, meaning that it's 50 years later than the first iteration of the verbal theory. It's a very, very long time before theory and formal modeling. And so as far as I can tell, this is the first kind of convincing paper about the quantitative genetics of robustness by Günther Wagner and colleagues. And so the idea is just to test the hypothesis about how robustness should evolve under classical quantitative genetic models. And the result of this and further work is that, okay, it's complicated. And the reason why it's complicated is that it's necessary to split the robustness into different robustness, independent robustness components. And particularly, it's important to be able to split the robustness to environmental factors and to genetic factors. Because environmental factors, they are not heritable by definition. They can be developmental or physiological, so they fit very well what Waddington was considering important about the canalization process. And it's about buffering the disturbances from the environment, while genetic factors are just heritable mutations, right? So one of the main issues with this split is that it would make Waddington very angry. This is angry Waddington. The AI couldn't generate a convincing picture of Waddington looking angry, so I just did it by myself. I'm not sure it's more convincing. So Waddington explicitly wrote several times that someone who wants to split environmental and genetic canalization was someone who didn't understand anything about the theory of canalization. So obviously he was completely opposed to this idea. So I'm not sure he would have liked a lot these kind of theoretical papers, and that's basically where theory splits between Waddington and modern quantitative genetics. So what happens is that, well, okay, we can consider a very, very simple classical quantitative genetics model in which the phenotype of an individual is the additive contribution of some kind of genotypic effect and environmental effect. So when under stabilizing selection, it's good to have a fixed phenotype, so it's good not to be robust now. But the question is how one expects robustness to evolve in such conditions. And so according to this theoretical framework, you could have different genotypes that differ by the variance of the environmental components. That's what we could call environmental robustness to environmental disturbances. So what happens is, in this case, if you have a small sensitivity to stochastic environmental factors, you are always at the optimal phenotype, so it's good. You have a good average fitness, everything is great. And when you have a large variance component, so sometimes you are still hitting the optimum, but most of the time you are off, right? And which means that if you have a large V component, a large variance of this E effect, then in average you have a bad fitness, right? So this genotype has a bad fitness in average, this genotype has a good fitness. And so there is a direct selective advantage of having a small V, and the selection coefficient is about of the order of magnitude of the difference in VE. So, okay, everything is fine. This guy should be selected, no problem with that. So why is it necessary to split environmental and mutational robustness? Is that with mutational robustness, the things are quite different. And the reason is that if you are mutationally variable, you still have the same phenotype. I mean, the offspring of these two guys will have different phenotypes, but these two guys themselves, they have the same phenotype. Both of them are optimal phenotype, and there is no fitness difference between these two genotypes, right? So what happens is that there is still selection going on, but it's indirect selection through actually the average fitness of the offspring and not the fitness corresponding to the genotypes. And in that case, it can be shown that the selection coefficient is of the order of magnitude of the mutation rate. Because of course, if there are no mutations, that all the guys look the same. And so the average effect of mutation, it just translates into an effect that is proportional to the mutation rate. And for most biologists, they would assume that the mutation rate is by essence something that is small, right? Which means that actually the selection coefficient is small. So just to summarize this theoretical background, one expects environmental robustness to be theoretically evolvable. It requires these complex G by E interactions, but we know empirically that it's not very difficult to observe them. It should focus mostly on traits that are under strong stabilizing selection, because the effect of the evolution of environmental robustness is proportional to the strength of stabilizing selection, and it scales with the environmental variance. While mutational robustness is theoretically evolvable, but it requires an G by G interaction, so epistatic interactions. But in practice, when you fill coefficients into the model, it's very weak. And so it should work only on traits that have a very large mutational target, because it's scaled with the mutation rate. And of course, I mean, most traits have a small mutation rate. So this is a big picture. So what I will try to focus on is mainly this part, because it's not completely obvious which kind of epistatic G by E G interactions could generate the evolution of mutational robustness. And so I apologize for people who have trouble to read 3D plots like this, because I have a couple of them. So what I just wanted to represent here is some examples of genotype to phenotype maps. So here on the plane, on the bottom part, you have genotypes. So of course the figure is so good that we don't see the Y2, but actually, so you have 2 dimensions of genotypes. You could consider it as 2 genes, for instance, that have a quantitative effect on the trait. And Z is a trait, right? So this is the additive, an example of an additive model that's completely the standard model in quantitative genetics. And the hypothesis is that the trait contribution just adds up, meaning that the genotype to phenotype map is completely flat, right? And so, for instance, you could imagine that you have selection for an intermediate phenotype, meaning that the population can evolve along this line here. It has the same fitness everywhere here. And when you look at wherever you are on this line here, the average mutational effect is the same, right? So it's an example where the mutational effect cannot change, so you cannot evolve genetic canalization in an additive genotype-phenotype relationship. When you have epistasis, you have a curved genotype to phenotype map. So this is an example of a multilinear genotype-phenotype map. So you still have this additive contribution, and you add some kind of epistatic factor here. And so in this kind of map, so you can imagine that under stabilizing selection, you can still evolve along this line that has the same phenotype. But now when the population evolves here, it changes the average mutational effect, meaning that if you are here, you have a small mutational effect. If you are here, you have a large mutational effect. And so evolving canalization, genetic canalization in this system is just traveling along this line here to get to smaller or higher mutational effects, right? So this happens through indirect selection because, of course, the mean phenotype remains the same, but then the sensitivity to mutations will be different. What I realized as well, and it's not completely clear from the literature, is that epistasis is necessary, but it's not sufficient to allow for evolution of genetic canalization, because you can have epistatic genotype to phenotype relationships in which the mutational effect cannot evolve. I mean, that's an example. So this one is completely symmetrical. It's a kind of Gaussian distribution of phenotype, actually. And then if you follow the line that gives always the same phenotype, it just goes around here. So there is epistasis, the genotype relationship, the genotype phenotype relationship is curved, but wherever you are on this line here, you have the same mutational effect. So this is an example where you have epistasis, but you cannot evolve genetic effects. So to be able to evolve different sensitivities to mutation, you need specific genotype to phenotype relationships. And then the question is, of course, is it realistic to expect that genetic canalization should evolve? I mean, which kind of GP maps can generate this curved relationship in which genetic effects should evolve, and under which selection conditions? So actually, you really need these three factors to explain the evolution of genetic robustness. You need mutations, you need epistasis, and you need selection, right? And it's really the interaction between these three components that can generate this pattern. So this is some simulation studies I ran with my former postdoc advisor, Thomas Hansen. And so we considered this this multilinear model of genetic architecture. So it's just an extension of the additive model with some epistatic factors here that scale proportionally to the additive effect. So there is nothing really fancy, but it's a great model because you can explore it mathematically, which is not completely easy with most models of epistasis. And we ran individual-based simulations here. We applied stabilizing selection, and we just looked at the evolution of mutational robustness in this condition. So what we track actually is the variance of genetic effects, and the lower the variance, the more robust are the phenotype, right? So this was just to confirm what was known from theory, that the evolution of the mutational effect scales with the mutation rate. And we tried simulations with 3 different mutation rates: very small mutation rate with squares, average mutation rate, or medium mutation rate with diamonds here, and very strong mutation rate with triangles. And this completely confirms what was expecting from the previous theoretical work, that actually you get the evolution of canalization, so lower mutational effects, only when the mutation rate is strong. So it really scales with the mutation rate. So what about selection? And there is something a bit disappointing. We tried different selection coefficients. So this actually, I didn't put the picture, but this conditions the width of this stabilizing fitness function, right? Stronger S means that the stabilizing fitness function is narrower, so selection is stronger. And so what's kind of surprising here when you didn't think about this problem before is that strong selection actually, so that the 100 case like triangles, it doesn't really help the evolution of canalization. And the reason is that, okay, on an epistatic landscape, you accumulate mutations by mutations and compensatory mutations to maintain the phenotype as it should be. And if selection is very strong, you can never have the first mutation that segregates enough so that you can have a compensatory mutation. So you're basically stuck, you cannot mutate at all. And meaning that actually you get the most evolution of canalization with intermediate selection coefficients, right? And as you can see, it's not very impressive how much you can decrease the average mutational effects, even when playing with the selection coefficient. And we did the same by setting different strengths of epistasis. So I should have explained that before. But actually, the epistatic coefficients were drawn in a normal distribution at the beginning of every simulation. So in this model, epistatic coefficients are fixed; they don't evolve. But still, we can fix them at the beginning of the simulations, right? And the more variance we give them, and the more epistasis there is in the system, right? So when we increase epistasis, well, it was not impressive neither, and we got the most evolution of genetic canalization with intermediate values of epistasis. Right? So just to summarize this, yes, indeed. So the evolution of genetic robustness is theoretically possible, but actually in practice, it's a bit disappointing because even in simulations where the simulations last for tens of thousand generations under constant stabilizing selection, which are the optimal conditions for the evolution of genetic robustness, then we don't observe that much evolution. So it's mainly driven by the mutation rate, for sure. This is completely confirmed, and other parameters don't have an impressive effect, especially because both selection and epistasis, I mean, there is an optimal intermediate values of selection and epistasis that maximizes the evolution of robustness, meaning that you cannot really expect some kind of extreme model species in which evolution of robustness can be extraordinarily large. It's just because, okay, it has to be medium to be optimal. So you don't really expect something very crazy to happen. So that looks a bit depressing in terms of, okay, is it realistic to expect the evolution of genetic canalization? And maybe one line of thought is that this is just because the model of genetic architecture is completely wrong. And the idea is that, okay, we took this very smooth genotype to phenotype relationship, meaning that close genotypes have also very close sensitivities to mutations, and maybe this is totally unrealistic. Maybe the sensitivity to mutations can evolve very fast because close genotypes can just differ a lot in terms of mutational robustness. What is quite impressive is that when you read Waddington, so Waddington was not a quantitative geneticist, and he was a developmental biologist, and he published a picture about what happens underneath the epigenetic landscape I've shown before. This is slightly less known, actually. So, and this is very modern in terms of representation, because all the pegs here, so you can see the landscape from below, right? And all the black structures here are genes, whatever. Okay, in the 1940s, I'm not sure that people had a very clear picture about what genes were, but actually these are genes. And these wires here, I mean, there are genetic effects on the landscape, and you see that they can cross each other and change direction. So this is clearly a representation of genetic interactions, right? And this is a very modern way of thinking about genetic interactions, as far as I can tell, because what it means is that, okay, let's imagine you have a mutation that removes one gene. What happens to the landscape? Where it mostly depends on the way these interactions, these wires interact, right? Maybe it can have a large effect, maybe it can have a small effect. And it's my understanding that in Waddington's mind, the structure of the genotype to phenotype relationship was really what we could call today a very complex genetic architecture with a lot of genetic interactions. And it really calls models based on the concept of gene networks and not these very smooth quantitative genetic models. So if I may reformulate the hypothesis, I mean, what would think that what happens is that genetic canalization emerge from complex gene networks in a similar way that consciousness emerges from the network of neurons, for instance. If you simulate one or two neurons, you never have consciousness. You need to have this very complex structure, and consciousness is an emergent property of that. So the idea that robustness might emerge from the complexity of gene networks. So meaning that basically, if you simulate a complex gene network, you might see things happening that couldn't be simulated based on very smooth genotype-phenotype maps. One of the reasons is that actually gene networks allow for feedback loops, and feedback loops are really important structures in controlling something, right? If you have negative feedback loops, you can just compensate for changes and maintain whatever you want to maintain without the need of something extraordinary. So gene networks allow for negative feedback loops, and which is not allowed by classical quantitative genetics. So actually, when I took my position in Paris, I developed a research program about that. So the idea was to generate a gene type phenotype model based on the principle of gene networks. And I picked transcription regulation as an example of this kind of complex physiological gene networks. Mostly because it has already been explored before, and we had good ideas of models that were simple enough to be implemented in computer simulations and still complex enough to carry this kind of possible emergent properties of biological complexity. And so the model is based on the idea that transcription regulation, so the expression of genes is controlled by transcription factors, and these transcription factors interact in very complex patterns that can be summarized by a matrix of interaction. So here you have the different genes, different transcription factors. These genes are expressed as proteins, and these proteins have an effect on the transcription rate of all genes of the network, right? So you have this loop here, and the idea is that it's a dynamic model. So for each individual during its development, you have a lot of feedback loops like this, and you end up controlling the expression of the different genes, right? That's the idea. So this model was developed by Andreas Wagner in the late 1990s. And now with modern computers, it's really possible to simulate very large populations for many generations. So what's elegant with this model is that there is a very clear split between genotype, which is the structure of the gene network, and phenotype, which is the expression of the genes, right? So you have to consider gene expression as phenotypes at the phenotypic level here. And the resulting genotype-phenotype map has specific features. It generates very strong epistasis, very strong pleiotropy, and non-continuous genotype to phenotype mapping, and it just emerges from this complexity. You don't need to add anything more to control pleiotropy or epistasis. It just comes along with the model, right? So I don't know actually if I should speed up or not, maybe a bit, but just to show you a bit how it looks like in terms of dynamics. So this is during the lifetime of an individual. It's a model with separated time. You have the evolutionary time in which you have successive generations, and within one generation you have every single genotype that develops into a phenotype. And so these kind of models can reach gene expression stability. So here it's a very nicely behaved genotype that just generates equilibrium gene expression that can be taken as phenotypes. But it's not necessarily the case. You can have something like this in which gene expression cycle in a very complex way. So it can generate very complicated phenotypic patterns. That's my point. And when you plot some genotype phenotype maps with the same representation I've shown before, so this is a phenotype. Okay, this is a phenotype here, and here you have two genotypic dimensions. You get a huge variety of possible landscape, from something that is kind of smooth to something that is very rugged, and you have these intermediate cases where you have very flat surfaces followed by cliffs. You generate all these kind of different patterns with a single model, right? So you have a huge diversity of genotype to phenotype mappings. So what we observed, we simulated these gene networks. We applied stabilizing selection on 2 genes out of the network. So we took very small networks with 6 genes to speed up simulations. I don't show the results, but it's exactly the same with larger networks. Actually, it doesn't really change the results. And so what happens is that we observe genetic canalization. So sorry, the axis is inverted compared to the previous figure. Like now, up is more canalized and down is less canalized. So in average, the network gets slightly canalized, but actually it's not very striking, and actually it's as canalized as controlled simulations in which there is no selection. So basically, this result is very similar to what we obtain with quantitative genetic results. We don't get more canalization with gene networks than compared to previous simulations. And okay, yeah, actually, I had this this figure with the network size. So, do we expect something emergent from this structure, like we tried with 6 genes? What happens to canalization when we have more genes? So actually, it's quite stable. So of course, I don't have direct evidence that something doesn't happen with 1,000 genes, but okay, it doesn't look very promising somehow. And in particular, the networks never get more robust than control simulation. So again, it's like, okay, stabilizing selection maintains some kind of robustness in network, but it's not very impressive. And there is nothing in the way regulatory networks work in practice that stimulates the evolution of robustness. So how does it work in practice? And so how do actually, when you look, one of the advantages of this kind of models is that you can look at what happens at the genotype level actually when robustness evolves. So we defined two different ways that robustness could evolve. The first one actually it's a kind of a trick by the model. The model tricked us because actually what happened in many simulations is that the mutational target shrinked. And the idea is that you can imagine this simple 3-gene network. So you have the blue gene that is under selection. So this one has to stay constant. And it's regulated by both the purple gene and the orange gene, right? And so it's actually the orange gene regulates the purple one that regulates the blue one. And so if you mutate the purple gene, then it will change the expression of the blue one. If you mutate the orange one, it will change also the phenotype of the blue one. But actually, if you lose this regulatory site here and you replace it with this green site, you can consider that the green gene is expressed constitutively, and so it comes from somewhere else. Then you lose the dependency to the orange gene. So this one can accumulate mutations without changing anything here. So that's a weird trick. The mutation rate was constant in this model, but still the network evolved so that the actual mutation rate evolved towards lower mutation rates. So it was a bit disappointing. And the other mechanism was redundancy. So this is kind of expected, but what happens is that just imagine you have to inhibit the blue gene. Well, it works better if you have two redundant inhibitors, because if one disappears, then the other one is still doing the job, right? And that's how we got robustness in this pattern. But unfortunately, it works only when genes are selected to be on or off, right? If you select genes to be expressed at intermediate values, it doesn't work anymore, because if the expression of the gene results for a complex balance between different transcription factors, if you remove one transcription factor, of course, things will change. So it's not a general mechanism. It works only with some specific targets for selection. So basically, this is why we did not observe very convincing evolution of genetic robustness in this network. So can canalization evolve in these complex gene networks? Well, not really convincing, because in spite of a huge diversity of genotype-phenotype relationships, implementing all kinds of smooth and rugged structures and so on, we didn't really observe something convincing about the evolution of canalization of genetic effects. So of course, robustness can evolve in gene networks, but only in specific situations and in a way that is not more convincing actually than in smooth quantitative genetics models. So nothing specific about gene networks here. Still, gene networks are specific, but the predictions do not differ really qualitatively from quantitative genetics models. And we couldn't identify any emergent property of these complex genetic regulations that would explain robustness in general. So to sum up a bit this part, okay, so the stabilizing selection can promote smaller mutational effects, so it's possible, but it's unlikely to matter a lot when you compare it with the strengths of other evolutionary forces like drift, constraining the genotype-phenotype maps, changing in strength of optimum during time, because of course it's very difficult to imagine constant stabilizing selection for tens of thousands generations long enough to generate this kind of long-term robustness. And you might have a lot of different mechanisms that might parasite the evolution of canalization. The main one is probably the evolution of mutation rate. It's probably much easier to decrease the mutation rate rather than to shrink the mutational effect. So if you need to limit the effect of mutations, it's much easier to decrease the mutation rate. So are we done? And I hope that given I'm a bit late, you were hoping that we are done, but not completely yet. Okay, so what about if we go back to the definition of canalization and we merge back again this problem of robustness to different kinds of disturbances? So what happens if robustness is a general property of genetic architectures? And so it wouldn't make sense to study only robustness to mutations. Robustness is unique somehow. So what might be that actually genetic robustness to mutation could evolve as a consequence of the evolution of robustness to environmental factors. If robustness is based on the same physiological mechanisms, then there is no reason to decouple both. And you would see genetic canalization to evolve even if it's not directly selected. So this is the congruence hypothesis that was raised actually quite soon after Günther Wagner's paper. And so the idea that actually robustness to mutations and robustness to environmental disturbances is the same thing. And so you observe actually different, you observe the same phenotype in different ways. And so genetic canalization just evolves as a consequence of environmental canalization, and there is just nothing to see in terms of evolutionary mechanisms that are targeting specifically genetic canalization. And okay, that's the same in terms of pictures, right? Genetic canalization evolves through that way and not through this way. So is it possible to model this using the same model? And actually, this is what I did recently. So the idea is to find different ways to disturb the system, to take the same model, but disturb it in different ways. So for mutations, we already know what to do is to change the genotype. So to change slots in this interaction matrix here. But how to change to simulate some kind of environmental disturbances? Well, the easiest way is to change gene expression, like saying that, okay, the expression of these genes can be disturbed by some environmental factors, right? So we disturb the phenotype, we keep the genotype unchanged, and we disturb the phenotype. Well, actually, it's a bit more complicated because we can change either the genotype or the phenotype at the very beginning of the developmental process or at the end, and it generates quite different patterns. So, for instance, early genetic mutations can be really interpreted as mutations in the germline. So the whole body carries a mutation, and all the cells are affected by the same disturbance. But if the mutation arrives late in the process, it's similar to a somatic mutation. So mutation that happens in some specific tissue quite late during the development. But what's possible to imagine that you might have selection on somatic mutations, like basically not getting cancer because it's bad for fitness. So it's not heritable, but still you might have selection on this that might have consequences on the robustness of the network in general, right? And one can do the same with environmental mutations. So actually, I have kind of different ways to disturb this network. And robustness was measured as the variance between, I don't know how many, like 100 independent disturbances. So the more variance you get into gene expression at the last time step, and the less robust the gene network is to disturbances, right? So my first question was whether robustness was variable and correlated, and different robustness components were correlated. So this is early environmental, late environmental, early genetic, late genetic, like different kinds of disturbances I've shown before. And the same here. And so I generated a lot of random networks and just estimated robustness and plotted the pattern, right? There is nothing fancy here. So the idea is that there is many orders of magnitude of differences between the different robustness. So these are log scales. So you have basically up to 10 log units in terms of variance due to depending on the network. So when you generate random networks, of course, it's not realistic at all. It's just to show a bit the properties of the system. And so there is a huge variation possibilities for robustness. And as you can see, all robustness components are positively correlated, meaning that if you are more robust to environmental factors, you are also, in average, more robust to genetic factors. But these correlations are never one. So there is still some room to change in different directions. So that was actually exciting because, of course, both are interesting, both the fact that they are positively correlated and that they are not 100% correlated. So I simulated artificial selection on robustness. So it's a purely theoretical experiment. Of course, you cannot do that in the lab. But the idea that what happens if you were able to measure the robustness of specific genotypes and select the more robust and the less robust ones for many generations. So these are log variances, so meaning that, okay, when it gets higher, it gets less robust. If it gets lower, it gets more robust, right? And for every robustness component, I added gene network stability here because it's correlated. It's not really a robustness component, but it's this ability, this trend for the network to cycle. And actually, it ends up being very correlated to stability because when it's cycling, it's less stable. But okay. And I simulated up and down response to directional selection, and the black dots here are control simulations with no selection. And actually, I got response to selection in both directions for all robustness components. So some appear to be asymmetric, but don't focus too much on it. It's just because, okay, it's more difficult to make the networks less robust, but this is a consequence of taking a random network as a starting point, because random networks are kind of not robust at all. So it's difficult to make them worse. But actually, there is a slight response to selection anyways. So this means that actually robustness is completely evolvable. It really behaves as a regular phenotype. If you could measure and select on it, it would respond to selection. And then I looked at correlated selection. So what happens if you select on environmental robustness? What happens to genetic robustness? And so this is the selected component, and this is the observed one. So on the diagonal, you observe the trait you select on, and off diagonal, you observe a trait that is correlated to the trait you select on. Right? And so what happens is if you select on any robustness component, you have a correlated response to other components. Right? So it's correlated. If you select for environmental robustness, you evolve genetic robustness. But you can also see that the response is less impressive than if you select on the trait directly. And this is because these traits are correlated, genetically correlated, but they are not perfectly correlated, right? So this is exactly actually the pattern you expect based on classical quantitative genetics models in which you have two traits that are genetically correlated. You select on one, you get a response to the other one because of genetic correlations. The robustness in this system, it really behaves as a quantitative genetics character. They are correlated, but there is room for evolution. So back to my original question, is environmental and genetic canalization the same property as what Waddington was considering? Well, okay. Unfortunately, it's complicated. And it's complicated because it's both yes and no. So yes, in the way that all measurements of robustness are correlated. So if you select on one, you expect a response to the other one. But no, because the correlation is not perfect. And still, every robustness component can have its own life. And I haven't shown the simulations here, but I tried to select on both in opposite directions. Like what happens if you select for more robustness to mutation and less robustness to environmental factors, and it responds. It's hard, it takes time, but you can decouple both robustness. It really behaves as correlated quantitative characters. So it's likely that if you might have correlated response, but you cannot exclude that each character, each robustness component follows its specific evolutionary trajectory. So I think that my main conclusion here is that free robustness behaves as a multivariate quantitative character, and you have to think about that when you think about evolution of robustness. So just to sum up, it seems likely that environmental canalization actually drives the evolution of canalization in general, because selection is direct on environmental robustness. So you might have a weak effect on indirect selection for robustness to mutations, but probably it looks likely that it should drive the evolution of the system should be driven by environmental canalization. So what happens to regulatory networks? Well, we succeeded in generating genotype-phenotype relationships that are extremely complex and that could allow any kind of evolution of robustness. But we could show that actually it's very complicated to make genetic canalization evolve in this kind of systems. So it's not like, it's probably not the way to go to explain why living organisms are robust in general. And the congruence hypothesis looks like an interesting track, because it seems unavoidable when you have complex genetic systems. It may explain the evolution of canalization in general, and it's still compatible with quantitative genetic models. So it puts everything together in a single framework. But still, these robustness components may remain partly independent. And so, for instance, things that remain as open questions is the question of, in general, pleiotropy and modularity. Like, okay, I've modeled only single characters. Like, okay, what happens if you look at on one phenotypic trait? But actually, the phenotype is multidimensional. So what happens to the phenotype in general? Can you canalize, for instance, one phenotypic trait and make another phenotypic trait less robust? Is it possible to do that? Or is robustness a general property of the system? What's the role of the trait correlation into the evolution of differential canalization of different traits? And well, that's a complicated question, I guess. And another very interesting thing is, of course, the evolution of phenotypic plasticity, because plasticity is a directed evolution of lack of robustness. You need to be sensitive to some environmental factor. And this, of course, seems completely contradictory with the idea of this congruence hypothesis that, okay, robustness is driven by the lack of robustness to environmental factors, because when you select for plasticity, you select for sensitivity to the environment. So is it possible to evolve both plasticity and robustness to mutations, for instance? I think that's really exciting questions for me, maybe not for everyone, but I feel that this looks contradictory with this theoretical background. So I'm quite excited by this kind of perspectives. Okay, so I'm a bit late. I'm sorry. I just want to thank all my collaborators for discussion and co-publication and many things. And of course, my students, so I haven't cited them directly during the talk, but actually all this work has been mostly done by students. So especially Estelle, Andreas, and now Apolline, who is finishing her PhD about evolution in a pleiotropic of robustness in a pleiotropic context. So, okay, this is, of course, all the others, and I thank you for your attention, and I guess that we have a bit of time for questions. So